Hello, my name is Arsh, and today we will be studying the topic of translation, the process of protein synthesis. This topic is in the chapter Molecular Basis of Inheritance, and it is a pretty important topic in the chapter as it is the final process of protein synthesis. And a lot of questions from this topic have been asked in previous board examinations and in school pre-board examinations, unit tests, etc. This topic is pretty important. We will also have to say that please, when you're studying the chapter, don't skip any topic because this chapter for the 2022-2023 batch had the highest weightage. I think above 10 marks out of 70. And even if you check the previous stats of which chapter had the highest weightage, this chapter had the highest weightage. On that chapter, this topic also had a huge weightage. Translation, transcription, these two are topics are pretty important topics because a lot of questions from these topics have been asked in previous board examinations. So yeah, don't skip them. Also, before we start the video, I wanted to say that when you're watching this video for the first time, don't write anything. So books, pens, pencil, keep them to the side and just focus on the video. Don't write anything. Don't listen to music or some people do that. Don't listen to anything and watch this video. Just try to give you full attention to this video and try to understand what's being taught. Like develop the concept. Don't just try to skim over the information given and just be like, okay, I can memorize this towards the end. No, that won't work. It won't work. And like you would if you're doing that, then you will have to skip a lot of portion, which increases the risk. And we don't want that because especially in biology because it's a really scoring subject. So yeah, don't do that. Just start to develop the concept by watching this video the first day, trying to understand how this process happens because it's a process, it's a mechanism. So if you understand it, that's more than enough. You don't need to memorize it. And also, if you are planning to memorize it, if you understand it conceptually more, then it will be very, very, very easy to memorize it. So in the first read, try to understand it. Second read, try to make your own notes. These notes are like fit for you. So don't be like, oh wow, my friend has notes for this, so I'll just read his notes. No, that's for him. Those notes he made for himself, so they're best suited for him and not for you. So in second watch, try to make your own notes, try to arrange topics however you feel like, use your language to explain it. And also because in this we have not included huge slides with a lot of like words, like we didn't copy from a textbook and just inserted it. So we just have diagrams. If you're gonna explain it, so whatever we are saying, sorry, whatever I'm saying, because I'm the only one here. So whenever I'm saying is try to understand what I'm saying and try to note it down in your own words. Because in boards you get marks for writing in your own words, but still, while making down notes, please hope not to miss any information which you might say. I will be saying topics on or things which are really important. So if you are sadly watching this towards the end of the year, then yeah, these are the things to focus on mainly. But if not, then congratulations. So far, and then yeah, just try to write on everything and don't miss anything. And also make sure your language is clear while writing on notes. So it's not like you just write few words and start to write whole sentences and yeah. So down the line, when you use your notes again, it will be good for you. So yeah. So we can start with the presentation. So what is translation? The process of formation of protein from mRNA is termed as translation. So, very simple. We want to make protein from mRNA, and that process, the whole process, right, the process of making that protein from an mRNA sequence, sorry, is termed as translation. Oh, also, just to inform that all this was handwritten, so we wrote this by hand because we did not have access to an iPad during the filming of this video. So yeah, we've tried to written down the notes by hand. Sorry for the sloppy handwriting, because this was written in Ramadan and I was fasting, so yeah, excuse me for that. But yeah, I've tried to write them neatly as possible. If there are still few words which are unclear, which could be the picture quality or just my handwriting, then I would be reading down the whole slide. So you might, if you see a word which you might not be able to read properly, then I will be reading it down. So then after that, you can read it and write it down. So yeah, please excuse this for this time. And yeah, we can start. We can continue. So central dogma of molecular synthesis. This is pretty important. Okay. So central dogma of molecular synthesis, it is proposed by Francis Crick. I think it's pretty important. A lot of things in the chapter were made by Francis Crick. So yeah, it's a pretty cool guy. And information is all. Next point. Information is always unidirectional, universal. So unidirectional means that from DNA you can get RNA, from RNA you can get protein. It's not that okay, I have protein, from this protein I can make mRNA, and from this mRNA I can make DNA. But okay, this process was reverse transcription, which happens in retroviruses, where suppose let's take the example of HIV, which it enters your body so that it enters in the RNA sequence, because viruses are RNA organisms. So this RNA converts itself into DNA, and then this DNA again forms RNA protein, and it infects the host. We won't go into too much detail. There would be a video on topic HIV which we made shortly after. So yeah, those videos you can just watch that for more information. But remember that the reverse transcription happens in retroviruses, and it is universal. So it means the process centered on biology is same for you, your friend, your friend's cat, your dog. Amoeba, the small microorganisms which you cannot see. You need a highly specialized microscope to see them. All the way to blue whales, elephants, birds, all these animals. They all have the same central biology, that they have DNA. This DNA makes mRNA and protein. There's no exceptions to this rule. Right, so eukaryotes and prokaryotes. Now, how does translation differ in eukaryotes and prokaryotes? Well, it's really simple, really. It's mainly differences in transcription process in both of them. So we'll just quickly go through that. In prokaryotes, only one type of RNA polymerase is needed for making RNA, mRNA, tRNA. No need of capping, tailing, and splicing. So, RNA polymerase, if you do not know, is basically the enzyme which catalyzes the process of transcription. Transcription, as we discussed below, is the process of making RNA, RNA from DNA, right? So yeah, so prokaryotes have only one RNA polymerase which does that process, and that RNA polymerase is capable of forming all three of RNA, mRNA, tRNA. So all these three made by one RNA polymerase, which is it's pretty cool. And moving on, we have no need of capping, tailing, and splicing. So if I hope you know what is capping, tailing, and splicing. If you do not, I'll just give a quick reminder. Capping is the process where the five prime end of the transcription unit we add a methylguanosine triphosphate unit, and tailing is the process where in the 3' end we add a adenosine triphosphate unit. And splicing the process where the hnRNA, which is composed of introns and exons, the introns are removed, only the functional exons are left. Because introns, it's non-functional, and exons are functional, so we don't want any non-functional members in the strand. So splicing, we can remove those. Moving on. In eukaryotes, different varieties of RNA polymerase are needed for making rRNA, mRNA, and tRNA. So in eukaryotes, these complex organisms, they require different RNA polymers to make their rRNA, mRNA, tRNA. So a particular RNA polymer sequence is used for making tRNA, a particular is needed for mRNA, and another for rRNA. We do need capping, tailing along with splicing, because yet again, because in eukaryotes we have DNA. That DNA is converted to hnRNA, or heterozygous nuclear RNA, and that is then converted to mature mRNA. So the hnRNA is immature; it is converted to mature RNA. Right. Moving on, different RNA polymerases. So examples. So RNA polymerase one. This is really important. This is really often MCQs. So RNA polymerase 1 encompasses rRNA, polymerase 2, mRNA, polymerase 3, tRNA. RNA polymerase 1 RNA, we have 28S, 18S, 5.8S, and 5 and 40 RNA. We have S5 sRNA and snRNA. These information in this bracket is not really important, but and no questions are asked from them. But if this question luckily comes to your exam, then in transcription, translation, any yeah, so you can add these 2 points and it'll put a really good impact on your invigilator. Just only write them if you're really sure. Like if you really remember, okay, 28 as 18 as 5.8 as. If you're like confused, like if you're not sure that okay, is it 28, 30 or anything else, then don't write it. Don't write whatever you know. Don't write anything which you're not sure, especially in your school exams, because then they will really cut your marks. But in boards, you might get away, but then it's not worth the risk, right? So, bonus fact: prokaryotes, the process of translation happens in cytoplasm, because they don't have a well-defined nucleus, and in eukaryotes it happens in the nucleus because higher organisms. So, how ribosome differs in prokaryotes and eukaryotes? Now, what is ribosome? Ninth grade, if you remember ribosome is a part of the cell which is responsible for protein synthesis. So, yeah, this is the protein factory. That's how we learned it ninth grade. This is the protein factory of the body. So, yeah. So, in prokaryotes, composed of 70 S ribosome. So S is the Schwetzberg unit. With and just no explanation. They won't ask you what is Schwetzberg unit, nothing. They will just ask you. They won't ask you anything. So, yeah. Just for your information, if you're wondering what is S is Schwetzberg unit. So in prokaryote it is 70S ribosome, and in eukaryote it is 80S ribosome. And in the prokaryotes, sorry, any ribosome is composed of two units. One is called the larger unit, and one is called the smaller unit. The larger one is obviously bigger in size, and the smaller one is smaller in size. So in the prokaryote, 70S is composed of two, 50S and 30S. This is 30, right? So yeah, this is 30. This might look like 80, but no, it's 30. That's the picture quality kind of looks better. Yeah, this is 30 right here. So 70S prokaryotes composed of larger 50S, smaller 30S. And eukaryotes composed of 80S ribosome, 80S composed of 2 units, larger is 60S, smaller is 40S. Right? Larger ribosome 2 units, larger, smaller, and so that. So, yeah. This is pretty important, this is actually really important. So yeah, if you again last minute learn this and go. Now, sections of ribosome, they have only three sections which we are interested in. We have A site, B site, and E site. So A site is the site of entry of a new tRNA. What is tRNA? We will discuss next time. So don't worry about it. So, yeah. So a new tRNA will enter from the A site, and B site has tRNA containing the growing polypeptide chain. So yeah. So okay. Uh, hope you know that a protein is a large sequence of amino acids. So yeah. And the P side, the building of the chain. So suppose this is one unit, and then another will get added, another one will get added, like that. Like that the chain goes. Yeah. This process of it growing, the chain growing, is happens in the A side. And in E side, it is the process through which the tRNA molecule, after delivering the amino acid, exits. So after the tRNA has done its job, it finally leaves the larger unit ribosome from the E side. Oh yeah, all these three sides, they're in the larger subunit and not in a smaller one. This is pretty obvious from random, but just letting you know. So we'll go through again. A site entry, B site synthesis, growing polypeptide chain synthesis happens here. And in E site, we have the amino acids exit happens to the E site. So easy to remember. E is exit, B is processing, A is okay. If this is A, this is exit, this is process, this has to be entry. Yeah. So yeah, if you're having difficulty remembering what is which, then yeah, E exit, B processing. The last one is A. So yeah. Components of protein synthesis. Yeah. Oh, yeah. Sorry. So this is the sorry. This is the largest subunit. Diagram. Largest subunit like this. Smaller is like this. Right. So yeah. If you're wondering how it, if you were confused here, this is how it is. Then in this the three sections: E side, B side, A side. So yeah. Translation unit. This is the RNA sequence from which we want, which has the particular group of protein which you want to make. So yeah, this is the unit which undergoes the process of translation, and the following components of this structure. It has first a start codon. The start codon is this one, AUG. AUG also codes for methionine, and it also has an initiator codon sequence. So yeah, start codon, AUG, the first one. After that, we have the region coding for the amino acid. So this whole region between, so this whole region, this is what we call. This is what we generally refer to as region coding for amino acid. This is the initiator region. You can understand this is the terminating region, this UAA sequence, and this little one which is flying by these two is the um, coding amino region coding for the amino acid. Then you have stop codon. Stop codon is located in the end. It could be UAA, UAG, UGA, any three of them. These all three don't code for any protein, and they help in the process of that is stop codon, and so they stop the process of transcription, translation. Yeah, so yeah, they're pretty useful because if you don't want this to go forever, that we won't get a protein if that happens. So yeah, these help in to when we dump the protein synthesis, they help to stop it. And we have something known as the untranslated regions or UTRs. These are really codons which don't code for anything. So yeah, okay. Now don't confuse stop codon with UTR. They are not the same thing exactly. Don't worry, no one will ask you to differentiate between stop codon and UTRs. So yeah, so you can just remember that the start codon, this sequence, and the stop codon, this sequence. These both are flanked by certain sequences which do not produce any protein, and these sequences are termed as untranslated region. So yeah, if they ask you to describe that, then that's the process. This is the definition you can go, or you can just say that codons which don't code for any protein. Moving on. Then we have review of tRNA. Okay, now we had discussed about tRNA, right? So it's a really major role. So considering that, we just decided to add a slide so we can review what is tRNA, and jog your memory about tRNA, right? So what is tRNA? tRNA, or transfer RNA. It is basically responsible for binding to the specific amino acid, and it brings the amino acid to the particular RNA strand which the process transition happens. So this tRNA, suppose this is amino acid right here. So what happened was this tRNA and this amino acid will join, and this tRNA. Suppose we have a structure unit over here, like let's assume. So suppose we have this one, yeah. So now for having translation, yeah, we need amino acids. So without them, we can't have. And then for translation, we have to bring amino acids from different points. So suppose from different parts of the body, we have to bring amino acids for the process of translation, right? So the process of bringing those amino acids is done by tRNA. That's what's called as transfer RNA. You can remember it. It is basically like the Uber of the body, where it brings the amino acids to this particular location for the process of translation. So the same way how your school bus might bring you to school for studying, this tRNA brings in the amino acids for translation process to the RNA sequence, right? Also, really important, the structure of this tRNA is really important because it could be asked to you in any exam to describe tRNA and what it is, what's its functions. So we won't go into the structure too much in detail. We will only look at the part of the structure which are useful for us. But yeah, this is very well discounted on that NCERT textbook, so you can just read once or twice, and that's that. So for what is relevant for us, we have the anti-codon loop. So anti-codon loops generally has the coding sequences which are complementary to the codon in the mRNA. So suppose we have AUG, so this sequence has UCA. So it has a complementary code for it. So because of this, it's able to bind to a specific strand of amino acid. If it did not have that, it won't be able to bind to it. Then on top over here, we have amino acid arm. So in this amino acid arm is the place where the amino acid joins, right? And also, important thing, remember that the 5' prime in this tRNA is towards your left, and the 3' is towards your right. So yeah, and these three can just read it to that as well. Now, steps in protein synthesis. Now, this has three steps: initiation, elongation, termination. Elongation is the main part of protein synthesis. But yeah, so we will begin now. So initiation. Initiation is a process which leads to the activation of the amino acid. So if you have to first of all make the amino acid potent, right, or capable of tons of the forming polypeptide chain. So we do and form it should be made competent from forming the chain, and also competent to join to the tRNA, right? So that we do it with the help of ATP. So there are two steps in this. So yeah, this you have to memorize anyway. So we have amino acid AA with ATP, which is adenosine triphosphate, also known as a currency of the body, which is the main source of energy. So AA plus ATP in the presence of amino acyl tRNA synthesis. I'll repeat, amino acyl tRNA synthesis. So AA plus ATP in this amino acid tRNA synthesis, we get AA-AMP enzyme complex. Please write this enzyme complex. It's really important. You might lose marks in your exam if you don't write this. They do look for these in your exams. If your luck is bad, you have a very picky teacher, then yeah, he might cut marks for you. Your marks if you did not write this. So yeah, please write amino acid AMP enzyme synthesis, right? Plus two phosphate group because ATP is triphosphate, so we have only one phosphate, so the two are left out like this. So now this is the first step. The second step, this AAMP enzyme complex plus tRNA, because tRNA is the cap, you can say. This will react, and this we get AA tRNA, which we wanted, plus AMP adenosine monophosphate plus enzyme. Now this is just uh, there's actually a huge process for this, but yeah, in our textbook it is simplified and it has enzymes. So yeah, don't be confused about it. just write down enzyme. That's all you need to know about this. There's no additional information for this. So yeah, we go quickly again. Initiation, process of activation of amino acid. This is done to make the amino acid potent to join to the tRNA and also for the process of polypeptide formation. The process of initiating two steps, where over here the amino acid joins to ATP and the presence of amino acid tRNA synthesis to form AA, AMP enzyme complex plus two phosphate groups. That's the first step. Second step is that where this AA, AMP enzyme complex joins with the tRNA and thus AA tRNA sequence forms and plus AMP plus enzyme. Don't forget to write this enzyme complex. Anyway, right. Now the next step, elongation. This is the step where we actually form the polypeptide chain, right? So this is pretty long. So yeah, try to understand this, very important. This is the part where students have confusion, right? So this is our unit of translation, the same one which we had. So this is the start codon AUG GUC next codon. So it's just an example, right? It's not like true, but this is just an example for us to understand. These are UTRs, right? So now for the beginning of elongation process, the smaller unit of ribosome will join like so. So this is a smaller unit. It will join below the transcription unit, right? So it will join like this. It will join in a way such that it houses the UTRS. The UTRS inside the smaller ribosome unit, right? So it's not like okay, it starts from the AUG GUC sequence like this. No, it starts from the UTR and goes to the second coding sequence. So next step is that after that is done, after small unit has joined, the first tRNA, the initiated tRNA. Oh, also, would like to say that this is the tRNA, the first tRNA. So this is called the initiated tRNA. But throughout the video, I will calling it one. So this is the first tRNA. After the second tRNA, third, fourth, fifth, for ease of explanation. So the first tRNA comes also called a tRNA. So the first tRNA comes has the complementary sequence to the AUG sequence and it has a methionine molecule because AUG methionine same codon. So it will come and will join to its complementary sequence. Now after it has joined to its complementary sequence like this, the largest subunit of the ribosome will come and it will join in such a way that it is in line to the smaller unit of the ribosome and that the first tRNA sequence is in the P site of the larger subunit. So again, we just go through quickly again. We try to go back as much as we can, so you're able to build up the process, right? So we had a sequence. The smaller unit joined. The smaller unit joined in such a way that it housed the UTR, the first sequence AUG, and the second sequence. After that, the first tRNA came, also known as the initial tRNA. It came, it had a complementary unit with the AUG, and it formed the bonds, and it also had a methionine molecule because the AUG codes for start codon and as well as methionine. And after this tRNA joined, the larger unit of the ribosome came because two units of ribosome came, and this larger ribosome unit joined in such a way that it was in line with the smaller unit. You can see it's completely in line to it. It's not like there's a bit ahead and this is behind. No, both large and small in the same alignment, and it also joined in such a way that the first tRNA is in the P side. So it joins in a way that the first tRNA is in the P side of it. Please keep in mind the first one is in P side, not in A or E. It's in the P side, right? So after it is in P side, yeah. Now the second tRNA will come, right? So another tRNA will come. Will have a complementary sequence to GUC. It will have another amino acid in its amino acid arms, and it will come and enter into the A site because A site is the site from where we enter. So yeah, so now we have the first tRNA at the P site, and now we have the second RNA in the second tRNA. So second tRNA at the A site and first tRNA in the P site. So when this happened, the amino acid from this tRNA it will be transferred into this new tRNA, right? So this methionine will be transferred into this sequence. It will be transferred because this is the process of polypeptide formation. So this amino acid is transferred to this new one. Now after that it has been transferred, the ribosome unit shifts one codon. So you can see that the ribosome was at this part, right? It was at this part. Now after this amino acid transfer has happened, it has moved this one. So now the E side is at the AUG and the P side is the GUC and the A side is the UGC. So it has moved one codon. Remember that. And now earlier, the second codon was at the A side. Now the second codon is at the P side. And earlier, the first codon was at the P side. The first codon was on the E side. So after this has happened, as it is codon TRN is the E side. If we just leave, it has done its job. It has bought us the amino acid, and it has done its job, and will go now. And another tRNA will enter into the A side. So yeah, this will happen again. So this will go, another one will come on A side. Now these two molecules of amino acid are transferred to the third tRNA, which comes on A side, and again it will shift one unit. And this will keep on happening until we reach the end, and we have a lot of amino acids joining, and this lot of amino acids form the protein which we need. And after this has come to the end, where the stop put on the UAA, UAG, UGCs, UAA, UAG, UGA. I hope I'm right. These sequences are on the A side. The process will stop because no tRNA forms a complementary, have complementary stands for this UAA because it's stop codons. There's no tRNA for them. So when this comes, oh, sorry for this mistake. It is a bit in out of line here, but just ignore this. These are all in line. So after this happens, this is missing, yeah. So there's no as not as not as the tRNA over here is missing, something else comes and joins to this UA sequence. It's called a releasing factor. Now this releasing factor, it liberates both the larger unit of ribosome, the smaller unit of ribosome, and also long polypeptide chain. So yeah, this releasing factor removes this ribosome unit and also this long amino acid chain, which is the protein which we need. So quickly, we'll just review it once more. Initiation for making the amino acid competent, A plus ATP amino acid tRNA synthesis, we get AMP enzyme complex for two phosphate groups. This AMP enzyme complex plus tRNA will give you AATRNA plus AMP plus enzyme. This is really important to mention enzyme complex. Please mention it. This you have to memorize because it's a complex name. Positive elongation, first step, the smaller the ribosome unit will join. It will join in such a way it houses the UTR, first codon sequence, second codon sequence. We don't mention first and second codon sequence, but yeah, just mention that it houses the untranslated region and also the first amino acid and second one. Like, don't mention it bluntly, but yeah, sort of, write in a fancier way, and after that, the tRNA will come and join to the AUG sequence right here. So the first tRNA joins to the AUG sequence, also known as initial tRNA. It has methionine. Then the larger ribosome unit comes in such a way that it is in line the smaller ribosome unit, and also the initiated sequence is in the P side. After this has happened, this as another tRNA called a second tRNA will come. It will enter the ribosome to the A side, and after this has entered the methionine from this P side. On the P side, first tRNA will be transferred to the tRNA in the A side, and after it has done, the ribosome unit will shift one codon instead of where the first tRNA is in the E side, the second tRNA is in the P side. And after the first tRNA has entered its E side, it has done its job and it exits, and the third tRNA sequence comes and enters the A side. And after that, the second tRNA, it transfers its amino acids to this one, and the chain continues to grow and move forward until we reach to the end where the stop codon is in the A side and the long polypeptide chain is formed. And over here, no tRNA joins to the UAA, UAG, UG sequence because these are stop codons. They don't code for anything. Instead, a releasing factor comes and joins to it, and this causes the release of the ribosome units and the formation of the long polypeptide chain. So yeah, that's all for the process of translation. I hope you understood it well, and if you have any doubts, you can comment down below. We try to answer. If you're not able to answer, you can ask your teacher. And yeah, the questions and everything is provided in the description, so you can just go and practice this. Watch this video for second time for the for making notes, and try to keep on revising your notes. It's a really important topic, so please don't skip it. Right? Even if you're studying late for it, don't skip this topic. Questions do get asked from this, and it's. I hope you found it easy. You hope you understood it. If you have any suggestions how we can improve our videos, please let us know in the comments. But yeah, other than that, there's nothing important. Otherwise, we could discuss in this topic. Everything has been discussed there in regard to CBSE, with in sensitive board examinations. So yeah, thank you everyone for watching. Have a great day, great week. Study well, have fun, and